Incomplete dominance is a form of intermediate inheritance in which one allele for a specific trait is not completely dominant over the other allele. This results in a combined phenotype. Let's say for a flower petal, capital R codes for red petals, while lowercase r codes for white petals. Heterozygous capital R and lowercase r genotype would result in pink petals instead of red petals. Because the big R allele does not completely mask this little r allele, both phenotypes are expressed. The ratio for the genotype would be 1, 2 to 1, and the phenotype ratio is 1, 2 to 1 as well. Here's an example. Here is our recessive and dominant phenotypes from our last video. With incomplete dominance, there's an extra player, a new phenotype, which is somehow a blend of dominant and recessive. Another good way to think about it is like a dimmer switch. A switch completely on would be dominant, while off would be recessive. Somewhere in the middle would be heterozygous. That illustrates the concept of incomplete dominance.
Another example of incomplete dominance is sickle cell anemia. When a person inherits two big S alleles for red blood cells, he has normal red blood cell. If he receives two little s alleles, he will exhibit sickle cell anemia, where the red blood cells are sickle-shaped and they don't carry oxygen as efficiently. If he receives heterozygous big S and little s alleles, he has unusually strong protection against malaria. Both the genotype ratio and the phenotype ratio are 1 to 2 to 1.
Codominance is when the dominant allele and the recessive allele are both expressed in a heterozygous genotype. For example, if a dog with black fur, let's say his genotype was capital B, mates with a dog with white fur, whose genotype is capital W, their offspring would have a mixture of black and white, not necessarily gray, but spotted like a Dalmatian. If the black W allele had incomplete dominance property, then the offspring would have gray fur. There is only one possible genotype and phenotype from this cross.
Here's an example. Here you can only see one phenotype. Let's call it coat. And we can go ahead and say that it is also dominant. Thus, the genotype is big C, big C. Here, you can only see the opposite recessive phenotype. Little c, little c. Let's call this one shirt. In codominance, you see both phenotypes. Thus, you observe both the coat and the shirt, not some odd mixture of two pieces of clothing mixed together. That would look something like this. Weird. Let's say the offsprings of the black and white spotted male mated with each other. Then there are three possible genotypes available with a genotype ratio of 1 to 2 to 1. The phenotype ratio is also 1 to 2 to 1. Now to introduce karyotyping. This process is for visualizing the actual chromosomes of an individual and thus making predictions on their genetic health. Karyotypes are a complete set of an organism's chromosomes. Human karyotypes include 46 chromosomes with 23 pairs. 22 of the pairs are autosomal chromosomes, while the last one is a sex chromosome pair. Autosomal just means not a sex chromosome. Karyotypes are often used to diagnose genetic disorders.
Here's a karyotype of a normal human male. You can tell that the Y chromosome is on the bottom right. This individual appears to be healthy without any abnormal chromosomes. You can tell he's a boy because it has an X and a Y chromosome.
As you should know, XY means male, while XX is female. The process of making a karyotype consists of, first, collecting a DNA sample from the specimen, either by blood, or in a baby's case, the amniotic fluid of the mother. You must then extract the chromosome from the nucleus, stain the chromosome, then match the chromosomes by its size under a microscope.
Here's a karyotype of an individual who has Down syndrome. As you can see, for chromosome 21, this person has 3 of them. This is called trisomy 21, and Down syndrome usually results in some mental retardation.
Turner syndrome is another genetic disease that can be seen through a karyotype. As seen below, this individual is missing one sex chromosome. They only have one X chromosome. Some symptoms of Turner syndrome include short height or an incomplete development.
And finally, another genetic disorder that can be seen on a karyotype is Klinefelter syndrome. This individual has XXY as their genotype.
And don't forget, go blue!